Hello everyone, my name is Josué David Hernández Varela. I'm a professor and a postdoctoral researcher in the Escuela Nacional de Ciencias Biológicas in the Instituto Politécnico Nacional here in Mexico City, Mexico. In this opportunity, I wanted to present one part of the work that we're making with the students in the laboratory related to the sustainable carbon quantum dots obtained from AGRO-WASTE for fluorescent bioimaging of a microorganism, and in this particular case, Escherichia coli. In resume, we can explain that the obtention of quantum dots have several experimental setups. However, the microwave setup is the very easy one, and we can use different agro-wastes, in this case garlic, skin, and coffee husk. And after the procedure that include a mixture with water, ethanol, and sodium hydroxide, and the time in the microwave, we can obtain these carbon quantum dots from the different parts. Also, we perform an UV measurement and a particle size measurement to obtain the values of the spectra and also the size of the materials. And finally, the most important part, that is the confocal assessment, in which we use a confocal laser scanning microscopy to observe a colony of these microorganisms after the control grow at 37 Celsius degree and 24 hours, and observe it under the 405 nanometers laser. And the second part of this analysis is the observation of the solution of the quantum dots using petri plates or petri dishes with bottom glass. This with the intention to obtain the fluorescence intensity of the nanoparticles of the quantum dots. As you can see in the preliminary results, we obtained that the UV spectrum for garlic skin and coffee husk have the typical spectra for this kind of material, with the absorption peaks around 320. However, the most significant part of the figure 1 is the daytime and the UV light images that you can observe in the inset, because as you can see, the solution before the UV light have a yellowish tone, but when we turn on the UV light, you can observe a clearly bluish, greenish fluorescence. Besides that, we can observe the histograms for the particle size analysis using the dynamic scatter light, and we can observe that we have very small sizes of the materials below below 5 nanometers. With this data, we can resume that it's possible to obtain, but under this very easy procedure using the microwave, carbon quantum dots for garlic and coffee residues. However, the small sizes alone, with some quantum confinement effects that will be explained below, are better when the size of the carbon quantum dots is smaller. That's the reason because we now on we are talking only about the coffee carbon quantum dots. Below this, we can find the figure 2, in which you can observe the lambda mode image obtained with the confocal microscope for both carbon quantum dots, garlic and coffee. Garlic in the top, coffee in the bottom. As you can see, the lambda mode is no more than different images at different wavelengths under the same laser excitation. In this case, 405 nanometers laser. And as you can see, not all the spectrum reflects the emission of this autofluorescence of the sample. That's the reason because we wanna compare in the graph that are in the right part that the UV that is the gray line have a very distinct spectrum, but when we compare this with the fluorescent intensity obtained from the confocal image by image analysis, it's possible to achieve that the quantum confinement effect is better for the quantum dots that are smaller because the peak is not too broad, as you can see. Based on this, we select the coffee quantum dots as of the quantum dots with better fluorescence intensity, and we dye or stain frothies from the E. coli sample that we make before. And as you can see, we observe the E. coli in light microscopy with very distinct bacillus shapes, and as you can see in confocal microscopy under the same light that we use for the assessment in the lambda mode, we can observe the dye of the microorganisms using this material. In conclusions, we know that we can obtain the quantum dots, but until now we didn't was capable or to use these samples because normally in the extraction techniques we use different solvents, for example acetone or alcohol, and that could be killing the microorganisms. In these samples we try to keep the pH very similar to water pH by cleaning several times the materials, and as you can see the sample present a very good fluorescence. In the end, we think we can do more experiments based on these bioimaging techniques in order to observe if it is possible to dye the same microorganism but using two different laser lines to observe the autofluorescence of this. Thank you so much for your attention. If it is possible to you, you can put different questions in the bottom part of this description box. Thank you so much. Bye.